System.
The circulatory system is responsible for more jobs than you know, and it does them by using only two hard-working divisions, the cardiovascular system and the lymphatic system. The first system we'll talk about is the cardiovascular system.
Section A, the cardiovascular system.
The cardiovascular system delivers oxygen and nutrients to cells and removes carbon dioxide and metabolic wastes from cells. The cardiovascular system is made up of the heart and blood vessels.
The heart acts like a pump, pumping blood through the blood vessels. Blood vessels are like the plumbing that carries the blood freely through the body.
Hi, I'm a blood collector. My name is Chuck. A lot of people try to describe serial killers as bloodthirsty. This is simply not true. Well, take me, for instance. I have no desire whatsoever to drink blood. I just collect it and store it in my refrigerator to be used at a later date as engine fuel.
When you take blood and you break it down, it's made up of two components. They are the plasma and the formed elements.
The difference between the plasma and formed elements is that the formed elements are cells and cell bits, while plasma is mostly water with dissolved stuff like proteins in it. So the formed elements are living blood cells, full of life and born to party. They're the erythrocytes, or red blood cells, and the leukocytes, or white blood cells. There's also the thrombocytes, or the platelets.
Erythrocytes carry oxygen, the leukocytes fight infection, and the platelets, which are actually cell fragments, these little buggers help with blood clotting. That means they help you stop bleeding when you're cut. All these formed elements are suspended in the plasma. Plasma is a non-cellular, straw-colored, sticky fluid that's 90% water.
Now you have to understand that blood does a lot of things. These things will be covered in greater detail if you take physiology. For now, just remember that blood is all about exchange. Exchange, exchange, exchange.
Blood's in charge of exchanging gases, hormones, nutrients, and waste between tissues. So blood is like a liquid cargo ship, carrying stuff to and taking stuff from tissues. Exciting, huh? Another thing that blood does is regulate body temperature. It does this by absorbing and distributing heat. But that's not all. The blood prevents infection with its white blood cells. And on top of all that, the blood also maintains proper fluid volume in the body.
Wow, the proper fluid volume. You don't know how important that is. Without blood, we'd be a dried up husk like the guy in my basement. And just think, we have so much blood, the normal amount of blood in the human body is 5.5 liters. That's 8% of our body weight. That's 5 times more than an average serving of cola.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes, or red blood cells, leukocytes, or white blood cells, and thrombocytes, or platelets. Red blood cells have no nucleus, therefore they do not divide, and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.
The hematologic system is responsible for many functions, including the transport of oxygen and nutrients to, and the removal of carbon dioxide and waste products from the peripheral tissue. Disturbances to the hematologic system are common in the older person, and often the symptoms are vague and attributed to normal aging. Anemia or decreased numbers of red blood cells can be the result of nutritional problems, drug toxicity, chronic illness, and metabolic disorders. As anemia is a sign, not a diagnosis, further evaluation of the older person with anemia is always indicated. White blood cell production can be affected by disorders of the bone marrow, lymph nodes, spleen, and thymus. When lymphocyte formation is impaired, the older person will have problems with humoral and cell-mediated immunity and will be unable to mount an effective immune response. Platelet adhesiveness increases with age, and older persons are at risk for the development of deep vein thrombosis and peripheral edema, especially after suffering a traumatic injury, in the presence of atherosclerosis, or with changes within the blood clotting factors. Anticoagulation therapy is often prescribed for older people, and, with proper administration, monitoring, and support, can be a life-saving intervention. Many functions of the hematologic system remain constant in healthy older persons, including RBC lifespan, total blood volume, RBC volume, total lymphocyte and granulocyte counts, and platelet structure and function. Average hemoglobin and hematocrit values decrease slightly with age, but remain within normal limits.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups, based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Okay, for section 12.3 we're going to talk a little bit more about immunity. We'll address nonspecific immunity, describe phagocytosis and inflammation, discuss fever and how it defends the body, talk about antimicrobial proteins such as complement, and compose one good overview of the sentence, the purpose and the mode of action of the complement system. So our second line of defense is designed to pick off anything that might leak through our barriers and their secretions. And they include phagocytosis, inflammation, fever, and the action of antimicrobial proteins. Phagocytosis is designed to have a select population of cells to patrol tissues looking for injured or dead cells or the presence of pathogens or particulate matter that doesn't support life processes. It ingests and eliminates these materials and takes tiny bits of that material, presenting it on the surface of the phagocytotic cell as a signal to our adaptive immunity that there is a pathogen that has managed to breach the first and second lines of defense. And so it's going to be those components of our adaptive immunity, the lymphocytes, that recognize that presented information, that are going to be stimulated to divide and create an army of cells that will only attack and destroy a cell displaying that particular antigen, whether it be a pathogen or an infected cell. Okay, and so we'll get into, when we get into specific immunity, exactly how that's accomplished. The three kinds of phagocytes that patrol the body include neutrophils, monocytes, and macrophages. The neutrophils are a type of granulocytes. The monocytes are the precursors to the macrophages, and the macrophages are tasked with patrolling the body to destroy any loose pathogen or toxin that it happens to encounter. Neutrophils react early in inflammation against bacteria and foreign material and damaged tissue. A high neutrophil count is usually a sign of a bacterial infection. It's the primary component of pus. Monocytes transform into macrophages and migrate out of the blood and into the tissues, where they're going to perform the task of destroying the pathogen wherever it might be encountered. Histiocytes live in certain tissues and remain there during their lifespan. These include alveolar macrophages and the Kupffer cells of the liver, and the dendritic cells of the skin. So they're basically stapled there. They can't leave that area. And again, the role is similar to the role of macrophages that can patrol a multitude of tissues in the body. There's also macrophages in spleen, lymph nodes, bone marrow, kidney, bone, and in a roundabout way in the brain, okay, in the form of cells that are called microglia. A phagocyte is a cell that eats foreign material or material that's the result of damage due to pathogenesis. Phagocytosis is the physical process of eating up the material, attacking and dismantling foreign invaders. Again, these will be pathogens such as viruses or bacteria or parasites. They can be isolated. It can be an isolated event, or it can be part of the inflammatory process. Phagocytosis involves chemotaxis, ingestion, the formation of a phagolysosome, the destruction of the pathogen, and the excretion of the breakdown products. The pathogen-associated molecular patterns are signals found on microbial surfaces that are recognized by phagocytes and other cells of the immune system. The molecules are shared by many organisms but are not present in mammals and serve as red flags for phagocytes and other innate immune components. Bacterial PAMPs include peptidoglycan and LPS, while viral PAMPs include double-stranded RNA, which is not something that's seen in a healthy cell or even in healthy extracellular material. Pattern recognition receptors are found on phagocytes, dendritic cells, endothelial cells, and lymphocytes. They recognize and bind PAMPs. The cells possess PRRs whether they've encountered the PAMPs before or not. Inflammasomes are PRRs found in the cytoplasm of phagocytes of innate immunity that recognize PAMPs once they've been internalized. The recognition leads to the release of signals that initiate and regulate inflammation as well. So here's a look at phagocytosis.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
Here's a look at inflammation.
The inflammatory response is an important nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
Okay. The material between many tissues is liquid. We call it extracellular fluid. And it comes ultimately from the blood supply via the removal of fluid primarily from the arterial end of the capillary bed. This influx of fluid dilutes toxins, fibrin clots, trap microbes, and prevent them from spreading, so that the neutrophils can come in and destroy the trapped invader. The neutrophils aggregate at the inflamed site and phagocytize and destroy bacteria, and the pus is the byproduct of this warfare. It's the accumulation of white masses of cells, liquefied debris, and bacteria. Pyogenics are found on bacteria such as strep, staph, gonococci, and meningococci, and they stimulate the formation of pus. Fever assists inflammation by elevating the body temperature. It's associated with allergies, cancers, and organic illness. Fevers of unknown origin include intermittent fevers greater than 101 Fahrenheit, lasting over 3 weeks, for which we can't find a cause, after 1 week of patient investigation. The body temperature is maintained at 98.6 by the hypothalamus. A low-grade fever is between 100 and 101 Fahrenheit, a high-grade fever between 104 and 106. They're triggered by pyrogens, which are substances that reset the hypothalamic thermostat. Exogenous pyrogens are products of infectious agents like viruses, bacteria, protozoans, fungi, endotoxin, blood products, vaccines, and other injectables, while endogenous pyrogens are liberated by monocytes and neutrophils and macrophages during phagocytosis, and these include IL-1 and TNF. Fever inhibits the multiplication of temperature-sensitive organisms like poliovirus, cold virus, herpes zoster, and systemic subcutaneous fungal pathogens. It impedes nutrition of bacteria by reducing iron. It increases metabolism and stimulates the immune reaction and naturally protective physiological processes, speeding up hematopoiesis, phagocytosis, and specific immune reactions, while at the same time creating conditions in which the pathogen does not find it conducive to reproduce. The benefits of fever make it difficult to treat or not treat. Slight or moderate fever in an otherwise healthy person should be allowed to run its course. Side effects of fever include tachycardia, tachypnea, lowering of the seizure threshold. Experts agree that high and prolonged fevers in individuals with cardiovascular disease, seizures, or respiratory ailments should be treated immediately. So again, this is a tricky situation, right? The fever is designed really to help because the fever is going to create conditions where the immune system can reproduce, but the pathogen not so much. However, if the fever goes unchecked and gets beyond that 104, 107 window, that can result in death, okay? And so we have to be extremely careful about dealing with fever. In young children, this is one of the reasons we try to avoid the use of aspirin to treat fever because of the possible side effect known as Reye's syndrome. Okay, here's a look at interferon and how it works.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
Here's a look at complement and how it works.
The complement system consists of a group of over 30 serum proteins that play an important role in both innate and adaptive immunity. Complement proteins participate in host defense processes such as inflammation, attraction of phagocytes to an infected area, enhanced effectiveness of phagocytes, and lysis of cells. Complement proteins are activated by a cascading mechanism. There are 3 pathways of complement activation: the classical pathway, the alternative pathway, and the lectin pathway. The classical and alternative pathways are demonstrated in this animation. The classical pathway requires interaction of antibodies with an antigen, usually on a cell surface. Following binding of antibodies to cell surface antigens, C1, which is composed of 3 proteins, R, Q, and S, attaches to the constant region, Fc, of adjacent antibodies through its C1q subunit. C1s then acts on C2, cleaving it into C2a and C2b. Then C1s cleaves C4 into C4a and C4b. C2b and C4b bind to the cell near the antigen-antibody complement complex, and C2a and C4a are released. This complex of C4b and C2b, called C3 convertase, has protease activity and cleaves C3 into C3a and C3b. C3b becomes bound to the cell surface, whereas C3a is released. C3b then absorbs to C4b2b to form C4b2b3b. This structure is called C5 convertase. The alternative pathway does not require the presence of antibodies. Activation begins with cleavage of C3 into C3a and C3b, either spontaneously or by a blood protein. C3b binds to foreign substances. Factor B, a blood protein, attaches to C3b and is cleaved into two fragments, BA and BB, by factor D, forming C3bBB, analogous to C4b2b. Both structures, C4b2b and C3bBB, are called C3 convertase. C3 convertase rapidly cleaves more C3 into C3a and C3b. C3 convertase is further stabilized by properdine, which allows another addition of C3b, forming C5 convertase.
Other antimicrobial peptides are usually short proteins of around 20 amino acids that are able to insert themselves into prokaryotic membranes and kill microbes. These include defensins, meganins, and protegrins. They also have an effect on other actions of nonspecific and specific immunity. Researchers are looking for ways to turn these antimicrobials into therapeutic drugs in order to give us an entirely new class of antibiotics that we could use in the fight against disease brought about by prokaryotic pathogens, specifically things like bacteria. And so here's a look at how some of these natural antibiotics work. They basically insert themselves into the membrane, compromise the membrane function, causing the cytoplasm to leak out, and that leads to the destruction of the pathogen in relatively short order. Okay, so in that way they mimic antibiotics such as gramicidin. Okay, that brings us to the end of this chapter. Remember to review this material, as we will be testing on it in the quiz and in the final and in your practicals as well. And I'll see everybody in class. I thank you for listening.